Welcome to Ask Prep, your trusted companion for NEET and JEE preparation. Today we will study about a very important topic of biology, which is DNA fingerprinting, techniques and forensic applications. This technique has revolutionized how we solve crimes, establish biological relationships, and even conserve endangered species. It is one of the most powerful applications of molecular biology, combining the precision of laboratory science with real-world human, legal, and environmental issues. So, let's dive deep into what DNA fingerprinting is, how it works, and why it is so significant. Imagine that every person carries a unique biological signature, a pattern that is distinct, unchangeable, and present in every single cell of the body. That unique signature is hidden within our DNA, the long molecule that carries all the genetic instructions for life. Even though the DNA of all humans is remarkably similar, over 99.9% identical, the small differences, scattered across the genome, make each individual genetically unique, except for identical twins. DNA fingerprinting is the technique that allows scientists to detect these differences and create a pattern or profile that can identify a person. The idea of using DNA to identify individuals emerged in the 1980s when a British geneticist named Alec Jeffreys discovered that certain regions in human DNA, called variable number tandem repeats or VNTRs, showed high variability between individuals. These regions are made up of short sequences of DNA that are repeated multiple times, but the number of repeats differs from person to person. By analyzing these specific regions, scientists could generate a pattern, much like a barcode, unique to each individual. This became the foundation of DNA fingerprinting. Now, let's talk about how the technique actually works. The process begins by collecting a biological sample that contains DNA. This can be anything from blood, hair, skin cells, semen, or even saliva. Once the sample is collected, the DNA is extracted from the cells using chemical and physical methods. The next step involves cutting the DNA into smaller fragments using special enzymes called restriction enzymes. These enzymes act like molecular scissors, cutting the DNA at specific sequences. After the DNA has been cut, the fragments are separated using a method called gel electrophoresis. This technique sorts the DNA fragments based on their size by applying an electric current through a gel. The smaller fragments move faster and travel farther through the gel, while the larger fragments lag behind. This creates a pattern of bands on the gel, which can be visualized using special dyes or radioactive probes. The result is a series of lines or bands that represent the DNA profile of the individual. In modern laboratories, more advanced techniques like polymerase chain reaction, or PCR, are used to amplify specific regions of DNA before analysis. PCR allows scientists to work with very tiny or degraded samples by making millions of copies of the target DNA regions. Today, short tandem repeats, or STRs, have largely replaced VNTRs in forensic applications because they can be analyzed more efficiently and provide highly reliable results. So, how is DNA fingerprinting used in real life? One of its most well-known applications is in forensic science. When a crime is committed, investigators often collect biological evidence from the scene, such as bloodstains, hair strands, or skin cells left behind on objects. By creating DNA profiles from these samples and comparing them to the DNA profiles of suspects, forensic scientists can determine whether there is a match. This has become an essential tool in solving crimes, convicting guilty individuals, and even exonerating those who have been wrongly accused. Beyond criminal investigations, DNA fingerprinting plays a crucial role in establishing biological relationships. In paternity cases, for example, DNA from the child, the mother, and the alleged father is compared to determine whether the man is the biological father. Because a child inherits half of their DNA from each parent, the DNA profiles can reveal the genetic relationship with a very high degree of certainty. This method is also used in cases of inheritance disputes, missing person identification, and immigration cases where family relationships need to be proven. In the field of conservation biology, DNA fingerprinting is helping protect endangered species. By analyzing the genetic diversity of animal populations, conservationists can design better breeding programs, prevent inbreeding, and track illegal poaching. For example, DNA profiles of ivory tusks or animal skins can be traced back to specific populations, helping authorities combat wildlife trafficking. Even in agriculture, DNA fingerprinting has found applications. It is used to verify the identity of plant varieties, protect intellectual property rights of breeders, and ensure the authenticity of food products. For instance, certain premium crops or livestock breeds can be certified using their genetic fingerprints to guarantee that consumers are getting genuine, high-quality products. For students preparing for NEET and JEE, understanding DNA fingerprinting is not just about knowing the steps of the process but appreciating the broader impact it has on society. This technique beautifully demonstrates how the principles of molecular biology and genetics can be applied to solve real-world problems. It connects laboratory science to fields as diverse as law, medicine, conservation, and agriculture. It also highlights the importance of scientific innovation, international cooperation, and ethical responsibility. To sum up, DNA fingerprinting stands as a remarkable example of how a molecular technique can change the world. It has transformed the way we approach justice, parentage, biodiversity, and genetic identity. As young learners and future scientists, you are entering a world where genetic technologies will play an ever-increasing role. Whether you go on to study medicine, research, law, or environmental science, the principles behind DNA fingerprinting will continue to be relevant and powerful. Keep exploring, stay curious, and remember that every discovery you study has the potential to unlock new solutions and new possibilities for society. Thank you so much for being a part of the Ask Prep community. We truly appreciate your time and dedication to learning, especially when it comes to mastering biology, a subject that's not only essential for exams like NEET and JEE. If you found this video helpful, please give it a thumbs up, share it with your friends who are also preparing, and make sure to subscribe to our channel so you never miss an update.